Hello and welcome to this poster presentation regarding rodent cholecystokinin-expressing interneurons in the basolateral amygdala, BLA, and how they control extinction expression. As you know, fear conditioning is a powerful model system for investigating neuronal plasticity and the mechanisms of associative learning, given that the basic underlying circuitry has been thoroughly studied over the last decades. In particular, the basolateral amygdala, BLA, subnucleus of the amygdala, is heavily implicated in auditory fear conditioning, wherein the pairing of a neutral tone, the conditioned stimulus, with an aversive foot shock, the unconditioned stimulus, induces learning-dependent synaptic plasticity, glutamatergic, sensory efferents, and local GABAergic inhibitory interneurons. Inhibitory processes are heavily involved in fear conditioning. For instance, the expression of inhibition-related genes in BLA is modulated by changes in inhibitory strengths during fear learning and extinction. Notwithstanding, research on the neuronal basis of classical fear conditioning has been mostly on the excitatory glutamatergic inputs on the principal BLA neurons. This focus has been largely due to the fact that fear learning transpires at these synapses explicitly. Therefore, the role of individual BLA inhibitory interneurons in fear conditioning, such as those expressed in the calcium-binding cholecystokin CCK protein, remains underexplored. Large CCKL interneurons in the BLA are involved in the modulation of fear extinction through endocannabinoid signaling. The activation of type 1 CP1 endocannabinoid receptors during extinction inhibits CCKL activity, potentially enhancing extinction learning. So our research question for this paper is going to be, what is the role of CCKL interneurons in the extinction of conditioned fear memory, and how does their optogenetic manipulation affect extinction performance? So we aim to investigate the role of these interneurons during extinction of conditioned fear memories through optogenetic manipulation on fearly behaving rodents. We hypothesize that disabling endogenous cannabinoid neurotransmission in the BLA by optogenetic activation is expected to impair extinction expression, evidenced by an increase in freezing percentage due to the US, the conditioned stimulus, during extinction memory retrieval. We're going to use 22 CCK cream mice, 11 per group, 10 female, 12 male, in this background, 12 weeks old, in this mixed 2x3 design, wherein the between subjects factor is going to be the random allocation. The two conditions, the experimental and control group, whereas the within subjects factor is going to refer to the levels of CS-US pairing without optogenetic manipulation, and optogenetic manipulation in the absence of CS, of any conditioned stimulus. Our variables are going to be, as usual, the independent variable and the dependent variable. The independent variable is going to be the presence or the absence of blue light stimulation during the CS-US pairing, and the dependent variable is going to be percentage freezing time, as scored by the ratio of freezing time per epoch to total epoch time. We're going to deeply anesthetize mice with thiamidormitor cocktail and place them in a stereotaxic frame, and small craniotomies will be made above the BLA with coordinates anterior posteriorly 2 millimeters, medial laterally plus or minus 4.5 millimeters, and dorsal ventrally 8.4 millimeters from the pregma. We're going to inject bilaterally 2 nanoliters per side at a rate of 50 nanoliters per minute. Of course, we're going to allow animals to recover for at least 4 weeks after viral infusion prior to any behavioral training or optogenetic manipulation to allow adequate opsin expression. In vivo imaging is going to, via fiber photometry, is going to optogenetically identify CCKL interneurons in the animals, in both animals, both control groups and experimental groups. For selective opsin expression in CCKL interneurons, the experimental group is going to be injected with an adeno-associated virus RAAV expressing the excitatory channel opsin 2 in a CRE-dependent manner under the control of TLX5 enhancer. Control mice will be injected with a CRE-dependent RAAV, which carries a flexed green fluorescent protein gene flipped in the presence of a CRE recombinants. This group of mice is going to be the control to demonstrate that any observed effects in the experimental group are due to the activation of CCKL interneurons by way of the CHR2, and not just the result of light stimulation or the expression of a foreign protein, which is why we do not opt for a sham lesion route. Now, let us move on to the procedure. All animals of both the control and intervention groups are going to be exposed to the following protocol. Day 1 is going to be habituation following the 4-week period of recovery. 5 presentations of the 2 distinct stimuli, CS1 and CS2, each lasting for 10 seconds. is going to be presented, the order is going to be counterbalanced. Animals will also be exposed to 5 repetitions of light stimulation used in subsequent conditioning in the absence of any CS to determine whether there are going to be baseline effects of opsin expression and freezing. Day 2, both CSs will be associated with it, be paired with a unconditional stimulus, a food shock, and CCK activity will be manipulated during the entire CS2-US pairing. CS1 will be within animal control, i.e. it is not going to be paired within light stimulation, whilst C2 is going to be paired with light stimulation. Blue light, in particular, of 443 nanometers to 100 millisecond pulses, delivered through optic fibers implanted bilaterally in the BLA to stimulate the CHR2 expressing neurons. Day 3, we're going to test for retrieval of expression memory. Both CS1 and CS2 will be presented 5 times, each without light stimulation. And the freezing behavior is induced by the control CS and the light paired CS will be compared to assess the effect of the optogenetic manipulation of the activity of CCKL interneuron classes for each animal. We expect that during testing, CCKL interneurons will significantly impair extinction expression, evidenced by an increase in freezing percentage to the light paired CS2 compared to CS1 and the baseline freezing. Here we have the mean percentages and standard error means respectively. Moreover, we expect the light stimulation to have no effects on control mice expressing only the green fluorescent protein. That is, control mice will show no changes in behavior or responses when exposed to light stimulation. This will indicate that the observed effects in light stimulation are going to be due to the fact that we activated the CCKL interneurons and not just light exposure itself. These results will be in congruence with the findings of Wolf et al. Regarding the role of inhibitory interneurons of the BLA in fear learning. We showed that optogenetic enhancement or suppression of parval booming and somatostatin expressing BLA interneurons enhanced or attenuated fear expressor learning and memory retrieval respectively. Moreover, we expect our findings to be consistent with pharmacological studies, which have reported a profound disruption of extinction memory expression upon administration of CB1 antagonists in wild type and CB1 knockout mice. In conclusion, our findings will be a contribution to the literature implicating a role for BLA inhibitory neurons in fear learning, specifically showing that facilitating the activity of CCKL interneurons inhibits the expression of extinguished fear memory. Now, infralimbic projecting cells strongly depolarize during fear extinction, thereby releasing entogannabinoids. Disabling this entogannabinoid signaling of CB1 receptors during fear extinction may therefore fail to suppress CCKL-mediated inhibition. Thus, the impairment that we observe in extinction memory expression with the activation of CCKL suggests that attenuating this activation could enhance extinction learning by disrupting the plasticity of inhibitory pathways. This will allow for the successful retrieval of extinction memories, offering potential therapeutic applications for anxiety and fear-related disorders, given that various forms of these disorders are partly based on the inability to suppress inappropriate fear responses. Our results will also allow for novel neuromodulatory interventions such as deep brain or transcranial magnetic stimulations. And these techniques could be refined to explicitly target CCKL into neural subclasses to help manage anxiety and fear-related disorders. Now, if optogenetic activation of CCKL induces poor extinction expression, the results of this study could inform pharmacological treatments aiming to suppress this CCKL activity. And this therapeutic intervention could be used in conjunction with exposure-based therapies to augment extinction learning and prevent the spontaneous recovery of fear memories in individuals suffering from anxiety disorders. Finally, our results may also have translational potential to clinical settings, given the success of CB1 agonists in enhancing the maintenance of fear extinction in humans. Thank you for your time and your attention.